Hello and welcome to this Synoptic Arctic Survey webinar addressing how a new Arctic Ocean is unfolding. My name is Øyvind Påske. I'm the chair of the Scientific Steering Committee of SAS, and with me I have Anne-Kari Meisingset. With us today we also have three experts who each will introduce various aspects of precisely how the Arctic Ocean is changing. Andrei Prushinsky from Woods Hole Oceanographic Institution will shed light on trends in the freshwater accumulation in the Beaufort Gyre and how it might impact circulation patterns and climate. He will be followed by Ryan Woosley from MIT, who will look at how changes in the freshwater balance is impacting the potential uptake of anthropogenic carbon. Marit Regstad from University of Tromsø, then Arctic University of Norway, will wrap up this webinar with a talk that explores the drivers and the potential fate of primary production in the Arctic Ocean. We encourage you who follow this to write questions and comments in the chat, which I will read out loud for the speaker to answer after they're done with their talk. By doing so, we're simply trying to save time in the interest of keeping up to the schedule and also saving you from the pain of switching and potentially messing up the flow of the webinar. I also want you to know that we're recording this session and that the recording will be made available on our webpage. If for some reason the webinar breaks down due to technical reasons, we will simply restart it and start over again. Hopefully that won't happen. Before I leave the word to Andrei, I want to inform everyone listening that the Synoptic Arctic Survey is now underway. This fall, two ships, the Japanese Mirai and the Korean Aran, are engaged in SAS cruises, and many more are scheduled for next year, partly delayed due to the COVID-19 pandemic. For those of you who are not familiar with the SAS, in short, it's a bottom-up initiative aiming to coordinate international research vessels in an effort to better understand the totality of change in the Arctic Ocean. The main target can be summed up in a single overarching scientific question. What are the present state and major ongoing transformations of the Arctic marine system? A comprehensive science plan and data protocols have been produced and agreed on, and I encourage everyone interested in SAS to visit our webpage and to study the science and implementation plan. And also feel free to contact us if you have questions. Sea ice has, since 2007, become the symbol of change in the Arctic. Back then, for those of you who remember, many of us were stunned at the range and the speed of change, captured in a single summer sea ice collapse. One reason why sea ice became a symbol of a changing Arctic ice apart is its visibility. Its absence is something everyone can fathom. Not as obvious, but equally large changes are taking place in the ocean, underneath the surface, where water masses and ocean life interact across a range of temporal and spatial scales. This budding reality warrants swaths of data, novel approaches, and creative minds. Today, we will, as promised, look at a few of the changes that occur in the Arctic. First out, as announced, is Andrei. I leave not the floor, but the window to you, Andrei. Please go ahead, and thank you for joining us.
Okay, thank you for inviting me to have a presentation about freshwater in the Beaufort Gyre. My collaborators are Rick Critchfield, John Toole, Marie-Louise Timmermans, Bill Williams, and Sarah Zimmerman. If you look in the climatology, you can see there is a salinity minimum in the center of the Canada Basin. It is very difficult to explain existence of this salinity minimum and high freshwater content. And in 2003, we formulated several questions and at least one hypothesis to explain this existence of this salinity minimum. So first of all, what is the origin of this salinity in the buffer jar? How does this salinity of freshwater content change in time? And what are the driving forces of the circulation? And how stable is the buffer jar system? So we assume that wind is the most important factor driving fresh water to the center of the buffered jar. Usually there are very strong anticyclonic winds in this area, and Ekman transport convergence in the center of the buffer jar, which results in the strong downwelling and freshwater accumulation in this area. So fresh water is collected to the buffer jar by Ekman transport from the 50 meters mixed layer. In summer, anticyclonic winds are weak or even cyclonic, and in this case, buffer jar releases fresh water to the rest of the ocean. So in order to test this hypothesis, we established buffer jar observing system, which consists of the moorings start here and sites of CTD and ship-based hydrographic observations annually. So moorings measure ice draft on surface layer currents, temperature, and salinity in the water column. We have sensors measuring CO2. We have bottom pressure recorder. And our observations already in 2003 showed that freshwater content in the buffer jar changed significantly in relative to climatology of the 50s and 80s. And in the 21st century, from 2013 to 2018, freshwater content reached maximum. So there is significant accumulation of freshwater in the area. Historically, we can look at how this fresh water can change. I mean, we don't have observations from these different years, starting from 1948, but we do have history of changes of regimes of circulation. We have alternating anticyclonic regime. During this regime, a buffer jar accumulates fresh water. And we have cyclonic circulation regime. At this time, Ekman transport diverge and freshwater content in the buffer jar has to be reduced. And you can see this blue bars show periods of anticyclonic regime and red bars periods of cyclonic regime. When we observe cyclonic regime, we observe great salinity anomalies. It was observed in the 1970s, in the 1980s, in the 1990s, when fresh water was released to the North Atlantic. But since 1997, we already have 22 years of very strong anticyclonic regime over the buffer jar, and during this time, ocean was accumulating fresh water. So this is an illustration of freshwater content in the area in the 1970s, and you can see distribution of salinity at different sections. And in 2017, you can see significant increase of freshwater content. Bar colors shown here. Significant freshening of the upper layer and significant downwelling, which results in these high numbers of fresh water content in this area. And this is how historically from our summer hydrographic surveys, fresh water was increasing from approximately 17,000 cubic kilometers in 2003 to 23.3 thousand cubic kilometers in 2018. You can see increase of red colors, and now they occupy practically entire area of our study. Interestingly, that in parallel with fresh water increase, Pacific water heat content was also increasing. And from 2003 through 2018, it was practically doubled. So if somehow this heat from the Pacific water layer released to the bottom of sea ice, a lot of sea ice will disappear. This is a very complex figure showing fresh water content time series from different instruments and methods. These black dots and black lines show fresh water content inferred from sea surface heights measured by satellites. Crosses show fresh water content monthly obtained from ice desert profilers. Red line shows fresh water content from model results. Bars show fresh water content from hydrographic surveys, which we do each year practically at the same time. And this line in the center shows fresh water content derived from mooring observations below 65 meters. You can see that moorings do not measure fresh water content in the upper layer because they start measuring of ocean properties starting from 65 meters. And this yellow line shows fresh water content in sea ice. You can see there is a negative trend in sea ice. It has huge seasonal variability in fresh water content, but in general, since 2003, at least 500 cubic kilometers of fresh water was released to the ocean from melting sea ice. Bottom diagram shows Ekman vertical velocity. So negative vertical velocities mean accumulation of fresh water. So through all this period, due to atmospheric circulation and sea ice conditions, Ekman pumping was negative and accumulated fresh water in the region except 2012, which was less anticyclonic than other years. So in general, from 2003 to 2018, Beaufort Dry accumulated about 6,500 cubic kilometers relative to climatology. If released to the North Atlantic, we will observe one more great salinity anomaly comparable with the great salinity anomaly observed in the 1970s. As I said, at the same time, we observe negative trends in sea ice drafts, and starting from 2007, ice becoming thinner and thinner, and old ice was practically disappearing. This is illustration how Ekman transport shown by arrows converge in the center of the Beaufort Dry in different years. Maximum accumulation was observed in 2007 because of extremely strong anticyclonic winds. Some fresh water comes from rivers, and major source for the Beaufort Gyre is Mackenzie River. And this is illustrated by these figures. Here is, you can see from city lights, flux of organic matter coming from the Mackenzie River, and it is diverted toward Beaufort Gyre. This figure shows model results and shows how these particles penetrate in the Beaufort Gyre in 2007. So concluding, we can say that there are two major factors which influence accumulation of fresh water and release also in the Beaufort Gyre. For accumulation, wind stress has to be negative. So circulation has to be clockwise or anti-cyclonic. At the same time, we have to have fresh water at the sources available to accumulation. So changes of these factors have to be in change. If we have significant Ekman pumping, we have to have fresh water to be available to increase freshwater content in the Beaufort Gyre. Recent studies also show that eddy fluxes can, as friction, can influence and limit freshwater accumulation. There are several papers about this, but we don't have time to discuss this in more details. Another conclusion that this really strong anticyclonic winds prevailing during these 22 years, fresh water from ice melt, Mackenzie River runoff, fresh water coming from Bering Strait, actually resulted in this unprecedented increase of fresh water volume in the region. Interestingly, that analyzing 2019-2020 wind forcing, we found that it was cyclonic, and we can speculate that in 2020 some fresh water was released from the Beaufort Gyre, and we suspect that, we hope that results of our 2020 hydrographic survey to the region will validate this hypothesis. Unfortunately, I don't have more time to talk about this, but we have Beaufort Gyre website where all data all available. A lot of results are available, and you can use it and read it. Also, we have two Journal Geophysical Oceans special issues. One was published in 2010, one was published in 2020. So in average, I think we published more than 200 papers if we take into account publications by people from different institutions and different disciplines. Thank you.
Thank you very much, Andre. We have a question from Mary Louise. Good to have you with us, Mary Louise. Thanks for the great overview, Andre. In what way does it look like the anomalously high pressure over the Arctic this year, I guess this summer in particular, is influencing freshwater accumulation?
This year, we observed low pressure system over the Beaufort Gyre. That's why we suspect that fresh water was released. So cyclonic circulation prevailed over the Beaufort Gyre. So that's why Ekman transport was divergent, releasing fresh water to the rest of the ocean. But we have to measure this in our September cruise.
Were you just answering Mary Louise's question now, or I got a little bit lost there? I'm trying to keep eye on the technical stuff.
Yes.
Okay, thank you. And so then there's the next question from Are Olsen. Do you have a sense of what governs anti versus cyclonic winds? And might these processes be predictable on annual to decadal timescales?
You know, if you look in our Arctic Ocean Oscillation Index, you can see that this periodical behavior of the decadal variability with alternating cyclonic and anticyclonic regimes was interrupted in 1997. And Dmitry Dukhovskoy showed that this is because a lot of fresh water was released from Greenland. So now we can expect 20 years of anticyclonic regime and 2-3 years of cyclonic regime. So probably next 2-3 years we will observe cyclonic circulation regime in the region. So, but there are no other ways to predict this, I think.
All right, thank you. There's another question from Rolf Gradinger. Thank you, Andre. If the freshwater is released, will there be an anomaly in East Greenland current in 1 or 2 years?
I think you will observe it in 6-8 months. At least some of fresh water will penetrate into East Greenland, and some of fresh water will go to the Baffin Bay and then return to the subpolar gyre in 5-6 years. But for this case, like it was observed in historically from great salinity anomalies analysis. So yes, this has to be registered in the Franz Strait and in the East Greenland current as well.
Excellent. There's another question from Maria Luisa Sanchez Montes. Thank you very much for the presentation. Could you please repeat what the freshwater accumulation in the Beaufort Gyre impacts the Pacific Ocean? How maybe?
No, Pacific Ocean influences freshwater volume in the Beaufort Gyre. Some freshwater penetrates through Bering Strait to the Beaufort Gyre with these Pacific waters. So there is about 1 Sverdrup of transport of water from the Pacific Ocean to the Arctic Ocean, and there is a significant volume of fresh water in this water coming from the Pacific Ocean.
So, and then there's, yeah.
So in this case, Arctic Ocean does not influence fresh water content in the Pacific.
And there's a last question here now before we move on from Philip Asmi. What is the relative contribution of sea ice melt and terrestrial river runoff for freshwater accumulation in the Beaufort Gyre?
So, as I said, we need fresh water in order to be accumulated. And we know that annually we have about 0.5 meter of fresh water on the surface of the Arctic Ocean from rivers. This is a huge source of fresh water. But a right wind is needed to bring this fresh water to the center of the Beaufort Gyre. So we can say that river runoff is a major source of fresh water. At the same time, we have fresh water from sea ice, but it changes seasonally. We have a lot of fresh water from sea ice, comparable with fresh water from rivers in summer and in fall. So if we have right wind, this water from sea ice will be pushed down in the center of the buffer gyre. But in winter, this fresh water will be taken when sea ice grows. So it depends on season. But in general, fresh water from rivers is a major factor contributing to fresh water of the Arctic Ocean.
Let's do a last question. It came up here now, but maybe keep it a little bit short, Andre, because it's a big question. But thank you very much for this presentation. Is there an impact on AMOC, on the Atlantic Meridional Overturning Circulation?
Theoretically, yes. Models, depending on their resolution, shows that there is an impact, especially if you have low resolution models. If you have high and very high resolution models, you will see that this fresh water will go along coastlines and will not reach regions of deep convection. So this is still open question.
Okay, thank you very much, Andre, and thank you all for asking very good questions. Keep them coming. The next talk is by Ryan Woosley from MIT, looking at the freshwater balance and how it impacts the potential uptake of anthropogenic carbon. Ryan, the window is yours.
Hi, everyone. Can you all hear me?
We can hear you good. Thank you.
Okay. My name's Ryan Woosley. For those that don't know, I'm a principal research scientist at MIT studying the marine carbon cycle and the uptake of CO2 by the oceans. And I'm going to talk about how the carbon cycle in the Arctic has changed over the last 20 years or so. And thank you to Andre, that gave a good background information for my talk. I'm sure you're all aware that the oceans take up CO2 from the atmosphere and that it's not evenly distributed. Instead, there's several hot spots. The most common ones we talk about are in the North Atlantic and Southern Ocean, because this is where deep and bottom water formation occurs. But there's actually significant uptake of CO2 in the Arctic as well. There's a lot of uncertainty in this uptake in the Arctic for two reasons. One, it's very hard to get there to make measurements. And two, it's a very complex environment. So it's a rather small ocean, about 3% of the surface area, surrounded by continents. And this results in a high proportion of continental shelves relative to its size and a high input of river water, as we've already heard, about 10% of the global river water. The circulation is also very complex. So you have Pacific water coming in from the Bering Strait, filling in the surface, and you have denser Atlantic water coming in and filling the deep basin. And the circulation is largely driven by bathymetry. So because of the complex bathymetry, you get these small basins. So also complicating things is, of course, sea ice. This is an image from last week showing in white the current sea ice extent and the orange line showing the median ice edge for the climatology. And you can see that there's been a huge increase in the amount of surface area of the Arctic. Yes?
Sorry to interrupt, but can you use the presenter mode?
So the Arctic is a relatively small ocean surrounded by continents, has a large proportion of continental shelves, and a high input of rivers relative to its size. It's also covered by sea ice, which, as we've already heard, has been decreasing. And so the idea is that as this sea ice retreats, there will be more surface area for the ocean to interact with the atmosphere and take up CO2. So through time, the amount of CO2 taken up by the Arctic will increase. And this is supported by measurements. So this is the surface PCO2 in from a cruise that I'm going to be talking about. And you can see that it's entirely undersaturated with respect to the atmosphere. The highest PCO2 was a little under 400, the lowest was around 150. And at the time, the atmosphere was around 405, 407 microatmosphere. And so there's a strong driver for CO2 to go from the atmosphere into the ocean because of this delta. Contrary to this is the idea that the Arctic is a rather stratified ocean. So this is a cross section showing the Pacific on the left and the Atlantic on the right. And it's characterized by a thin polar mixed layer. Below that, you have a halocline layer with a residence time of around a decade, overlying an Atlantic layer with a residence time of a few decades, and then the bottom water with residence time of several centuries. And so because of this stratification, it prevents or inhibits the carbon dioxide that's been taken up in that mixed layer from being transported to the interior. And so this will inhibit the amount of CO2 that the Arctic can take up, because not only is it not being transported to the interior and being sequestered, but new water can't come in to take up more CO2. So to see how the Arctic has been behaving over the last 20 years, we had a cruise in 2015, the US Arctic Geotraces cruise, which went from Dutch Harbor, Alaska, up along the Makarov Basin to the North Pole and back through the Canada Basin. And this was specifically designed to be a repeat of two prior cruises, one in 2005 in the upper right in the Canada Basin, and one in 1994 in the bottom in the Makarov Basin. And in the colors, I'm showing the total alkalinity in the surface water. And what struck me about this is that you can see a huge decrease in the total alkalinity or buffering capacity of the water since 1994. To set up the conditions of the sea ice at the time, so the light blue shows the sea ice extent in August of 2015 during the most recent cruise, and then the white shows the extent in 1994 on the left and in 2005 on the right. So you can see there was a large increase in the surface area of the ocean that was exposed to the atmosphere, which would indicate the chance for more CO2 uptake. What actually happened was much more complex than that. So I'm only going to talk about the Canada Basin section, but the Makarov Basin was very similar. So on the left, I have the measured change in DIC from 2005 to 2015. So the surface 75 meters or so saw huge decreases in DIC, 50 to 150 micromoles per kilogram. And then below that, there were small increases. And as I said, for the alkalinity, the changes were very similar. The upper 75 meters or so had decreases of 50 to 100, 150 or more micromoles. And then below that, in that halocline layer, there were little to no change in alkalinity. And so how does this affect uptake of CO2 from the atmosphere? I won't go into the methods too much, but I'm happy to take questions. It's an ensemble extended multilinear regression technique, which is just a statistical way of removing out the natural variability to leave the amount of uptake of anthropogenic CO2 from the atmosphere. And so I call this gas exchange. And what we found is that the surface actually took up little to no CO2. And in the intermediate halocline layers, we saw low uptake of 5 to 15 micromoles. Typically, what we would expect for this 10-year period is increases in DIC of 10 to 20 micromoles. So it was on the low side. And so what we think happened is that even though the sea ice extent was much slower, there wasn't very much uptake from the atmosphere. Rather, most of the uptake occurred in the surrounding seas, and that then got transported into the interior in the halocline layer. And because colder water takes up more CO2, in the winter there was more uptake, and so that got transported deeper into that halocline layer. So if we take the change in anthropogenic CO2 and integrate it to get a column inventory, so the total amount of CO2 in the entire column of water in moles per meter squared per year as a function of latitude, the mean is the red line, and then the blue shading shows the standard deviation. So for the Canada Basin, it was very low, and even the sign of the uptake was within the uncertainty. And the Makarovs, the uptake was a little bit more, but still rather low. How does this compare to other basins? At the same time in 2015, there was a German Arctic Geotraces cruises covering the Eastern Arctic. And they found that in the Eastern basins, the column inventory was about 0.5 to 0.7. This compares to a global average of about 0.5, meaning that the Eastern Arctic seemed to be about average in terms of CO2 uptake over the last 20 years, while the Western Arctic was very low. And to put this into more context, the North Atlantic has values ranging from 1 to 2. So this is almost nothing compared to what the Atlantic typically takes up. And so what is causing this? So on the left, I have a figure I already showed of the change in anthropogenic CO2. But the Arctic, unlike the other oceans, is going through a lot of other changes. So I already mentioned the change in TA, but there was also a large increase in temperature, larger than we see in the other ocean basins, and large decreases in salinity. And so when we take those factors into account, we get the figure on the right, which shows decreases in DIC of 50 to 150 or so micromoles in that surface layer, and then in the halocline layer, small increases of 5 to 10 micromoles. And if we compare this again to that measured change, it matches very closely. And so from this, we conclude that changes in total alkalinity are the driver of changes in the carbon cycle in the Western Arctic. So we've already heard a lot about these changes. So where are they coming from? So this represents the amount of fresh water along each of the sections. So 2015 is in blue, 2005 is in red, and 1994 is in black. So the solid lines show the changes in river inputs. So across both sections, there were large increases in river inputs, as we've already heard, indicating that that's the main driver of this change in alkalinity. River water in the Arctic has a flow rate rated average of around 1,000 micromoles, while the seawater has a value of around 2,000. So sea ice, which is shown in the dotted lines, changes were much more variable. Some areas had increases, some decreases, and the overall change was much smaller. And so here, a negative value means net sea ice formation, so more ice formed than melted in the region. And so what does this change in freshwater and alkalinity have on the carbon system? So we have DIC versus PCO2. And this black dot here on the right represents the conditions in 1994. And so if everything stays the same, but we have a little bit of warming and uptake of CO2 from the atmosphere, we get this red dot following the orange lines, and an uptake of CO2 of around 13 micromoles. If instead we have dilution by sea ice melt, we get a huge decrease in PCO2. And this results in a larger difference between the atmosphere and the ocean, driving an increase in the uptake of CO2. So you get around 35 micromoles of CO2 uptake. By contrast, if that dilution is caused by rivers, which have a variable alkaline, or a medium amount of alkalinity, so around 1,000, sea ice is around 200 or 300, you get a slight increase in PCO2 and a decrease in the amount of anthropogenic CO2 that the ocean takes up from the atmosphere. And the exact number for rivers varies based on the ratio of the alkalinity to DIC. So this is kind of an approximate value. But this value matches up closely with the measurements indicating that it's changes in river input that are affecting the uptake of anthropogenic carbon in the Western Arctic. And so I will just conclude, the Western Arctic over the last 20 years had below average uptake and storage of CO2. This was due to increases of river input, reducing the buffering capacity of the water and decreasing the uptake from the atmosphere. And the net result is a decrease in DIC in the surface waters. And of course, what changes in freshwater will happen in the future, we don't know. So we don't know if this will continue or if there will be a rebound in the future if the amount of fresh water changes. So I'm happy to take questions.
All right, thank you very much, Ryan, for that great talk. I have one question for you from Are Olsen. Have you looked at the impacts of this freshening on ocean pH and on calcium carbonate saturation states?
Yeah, so ocean acidification is very hard to look at because pH measurements are hard to make. They didn't make any in 1994, and then we can calculate the value from DIC and alkalinity, and then compare them to the measured values in 2015. And what we found is that the rate of pH decrease was about double the global average. So even though the waters aren't taking up as much CO2, they're still acidifying at a faster rate.
Okay, so we have another question from Toby Tyrell. The changing hydrological cycle is altering global salinity patterns. Have you thought about possible global implications of this for ocean carbon storage?
That's not something that I've really thought about. It's certainly we can see how fresh water has a large impact on the carbon cycle, and so the implications are quite broad. So for the other ocean basins, when we do similar types of analysis, we don't find changes in salinity to be significant enough to impact the carbon cycle. But I think that's certainly a possibility in the future.
Excellent. And from Rubao G, nice talk, Ryan. How the changes in biological productivity due to changing stratification regime and light condition contribute to the CO2 uptake in the Arctic?
Yeah, so that's another hypothesis that you can, as the sea ice retreats, you can get a big bloom of phytoplankton, which will draw down the PCO2 and allow for an increased exchange with the atmosphere. So this technique that I've used assumes that a change like that is kind of natural variability. And so this technique doesn't actually account for those changes if it's an anthropogenic change. But that's something that certainly can contribute to changes in the CO2.
Okay. From Rolf Gradinger, thank you for the presentation. Have you considered a change in biological processes affecting the carbonate system, or is this second order?
Yeah, this is very similar to the previous question. Certainly changes in calcification rates. So if as the omega or the saturation state decreases, you could get changes in organisms that produce calcium carbonate shells, as well as
To find the increase of DIC in the region.
activities, maybe an input of nutrients, that gets accounted for as a natural variability in this. But certainly those changes aren't all natural.
Okay, then we'll end it there. Thank you again for a nice presentation, Ryan. And thanks also to people for asking questions. Very nice. Then I think we can move on to the final speaker of this webinar. We're a little bit ahead of schedule, which is good. This is Marit Reigstad from University of Tromsø, and she will look at the drivers and the potential fate of the primary production in the Arctic Ocean. The window is yours, Marit, and I also encourage you to put the presentation in full screen mode. Yes.
Thank you very much, and thank you very much for having the presentation or the opportunity to present here today. And thanks also to the other speakers for very nice talks that in many ways provide important background for the ecosystem and the production. I will focus on the primary production of the emerging new Arctic because that is base of the food web. It's important for the ecosystem, and it's also closely connected to the physical and the chemical conditions in the ocean. And I will present some of our recent findings, not only from my own research, but more in general, what we have an overview of, both regarding the driving mechanisms and the potential fate. The new Arctic is already here. We see that there are less multi-year ice and there are more first-year ice that extend the seasonal ice zone where biological production is high. The old seasonal ice zone, seen in orange on the figure here, shows the difference between the winter ice and the summer ice, and it was mainly covering the Arctic shelves. The new seasonal ice zone is including both the yellow but also partly the orange areas, and we now have a seasonal ice zone where sea ice is present in wintertime but removed and melted in summer, which extend far into the ocean basin. And these open water areas facilitate potential production. And there have already been quite a lot of research, especially using remote sensing, that have addressed that question. And Lewis and his co-workers have recently published a paper where they have looked at the data from 2098 to 2018. And they show that there is an increased primary production in terms of increased chlorophyll A biomass that can be seen in most of the Arctic areas. But what they also show is that there is a high regional variability. So looking at different areas of the Arctic show that there are different patterns, as we also have heard in the previous talks. And what they figure is the strongest changes can be found in inflow shelf, both on the Atlantic and on the Pacific side. And they have addressed a bit why is it so, and then we move over to key drivers of Arctic productivity. So still referring to the work by Lewis and his co-workers, what they figure out when they were comparing the net primary production estimated from chlorophyll in the lower panel with patterns in chlorophyll A in the mid panel and open water area over the entire period from 1998 to 2018, was that the first decade, the net primary production was correlated with an increase in the open water area. While the second period, there was not a strong increase in the open water area, but it was a strong increase in the biomass of chlorophyll A. So they suggest that there are a shift in what are the main drivers of the increased Arctic Ocean net primary production. They discuss whether this can be related to increased growth, increased mixing, bringing up nutrients, or reduced grazing, but suggests that the advection of nutrients into the inflow shelves, where the increase is strongest, is a main cause for the increase in the second part. While the first part, it is the decrease in sea ice opening for a larger area where primary production can take place. So we move from a light to a nutrient limitation, and advection turns out to be an important process for this pattern. Advection can also be important transporting biomass. A recent study by Maria Vernet and co-workers suggests that not only nutrients, but also the algal biomass is advected into the Arctic Ocean. Again, this is the European Arctic and the inflow of Atlantic water, which is investigated. And the advection makes an important contribution compared to the local production, actually ranging 5 to 50 times the local production. Also, Osiel and his co-workers show that advection plays an important role, facilitating the northward expansion of more temperate species like the Coccolithophorid Emiliania huxleyi. So with a stronger advective component in the Atlantic water, these more temperate algae species are moved further into the Barents Sea and further north in warm years compared to cold years, and that is bringing these algae further into the Arctic. But also the vertical structure and the physical structure of the water column matters. Nutrients can also be supplied from below after winter accumulated nutrients have been consumed during spring bloom. And in the figure to the right, we see data that has been compiled by Randelhoff and co-workers on available turbulence measurements combined with nitrate flux from the Arctic Ocean. They find that with a weaker stratification on the Eurasian and Atlantic sides compared to the inner part of the Arctic basin and also more towards the Pacific, there is a higher nutrient flux on the Atlantic side. We could expect also, and it is likely that there is a strong nitrate flux, or we know it is from measurements on the Chukchi shelf and the shallow regions on the Pacific inflow, but there is no turbulence measurements that could be shown from this area. But it shows that the physical conditions, both through the advection and through the mixing and stratification, plays a major role in facilitating nutrients and also the algal bloom and the chlorophyll biomass that we see. In addition to these physical drivers of blooms and the seasonal succession as we know it, several studies point to changes in bloom patterns, that we actually have to change our paradigms of blooms. Ardina and co-workers pointed in 2014 to the fact that there has been a strong increase in number of double blooms, meaning that we see autumn blooms that has been facilitated by delayed freezing of sea ice in autumn. Osmi and colleagues have shown how the more frequently appearing leads in the thinner and more fragmented sea ice act as windows to the under ice algal communities when they refreeze without snow cover, and that this is facilitating under ice blooms. And in the paper from 2017, they show that within a short time, there was a development of a Phaeocystis bloom seen in the picture to the right over about a week from a very transparent blue water to a green water dominated by algae. And this was facilitated by light coming in through the leads. And Fernandez and colleagues show that snow infiltration blooms, earlier known from mainly Antarctica, now is also seen in the Arctic when heavy snow push thin ice underwater and facilitate a habitat for algal growth on top of the sea ice and under the snow. So one of the needs we have for the future is a better observation of these new algal bloom patterns. And then we move a little bit to give an example on potential fate of the Arctic productivity. And I will also here provide example from the Atlantic inflow region where I have been working most. And we have the ecosystem includes a lot of different components, organisms living both within, under the sea ice and on the seafloor. But the algal bloom is providing food for the entire ecosystem. And because of that, we'll here zoom in on the increased algal biomass. Algae is not only algae. Algal groups can vary in size with a factor of 1,000 or more. As we can see in this upscaled picture here. And depending on the size of this algae, the fate of this production can be very different. The algae form planktonic communities where you have food webs that link not only the small organisms in the plankton community, but also the particulate and the dissolved pools. Large algae are favored by high nutrient supply and tend to feed larger grazers, while small algae have a competitive advantage if the nutrient concentrations are low, and they are suitable food for smaller grazers. Over the past years, we have learned quite a lot more, about these algal communities and the microplankton and microbial community due to molecular screening. So we are getting a far better understanding of the communities we find, the function and the succession. And what we find is that the Arctic communities are not that different from what we can see in areas further south. But an important issue to establish when we want to explore fate of productivity is who are the grazers, to establish interaction among the organisms. Larger copepods like Calanus, here exemplified by Finmatricus, but also Glacialis and Hyperboreus, which are larger congeners of the species, tend to dominate in biomass. And in this example, from the shelf northwest of Svalbard in the seasonal ice zone from 2014. In August, they made up approximately half of the biomass, with microzooplankton making up the other half. But Svensson and her colleagues, they also made an estimation of the ingestion that these different groups had based on their metabolic needs. And what they could see was that the microzooplankton was clearly dominating when it came to ingestion. And it's a well-known fact that small organisms have a higher production per biomass ratio, but that really plays a big role and have a big impact when we talk about food web and energy transfer in food webs. This was not only a phenomenon observed in August, but also in May, grazing from microzooplankton turned out to be very important. And the algal size and the grazers can fuel a shorter or a longer planktonic food web. And this has an impact of how efficient the energy is transferred. Larger algae can feed larger zooplankton and supply a shorter and more efficient food web because the energy move only through fewer trophic levels, and more of the energy is transferred from one organism to the other. While small algae and also together with bacteria, they can supply a longer microbial food web that are efficient in recycling nutrients, but they have higher metabolic losses, being more trophic levels. All ecosystems compile both of these types of food web, and they interlinked, and it's not as separate as we see it here. But one component can be more prominent than the other in different ecosystems and different times of the year. And an important question is, do the increased primer production in the Arctic that we see fuel the more classic or the more microbial food web? And that matters for whether we are going to have more energy towards, for instance, harvestable production like a fish community, or if we are actually fueling a more regenerative food web dominated by small microplankton. Another important aspect when we talk about both ecosystem but also relating to the physical and the chemical conditions is to understand regions and how they connect through the Arctic. I lead a project called the Nansen Legacy, where we have focus on the Northern Barents Sea, and we have an aim to provide interdisciplinary regional knowledge on the Barents Sea, especially the north, which is seasonally ice covered, and we use a climatic gradient through the ocean to establish this. It's a 6-year project, and we are in our 3rd year, so there is a limited amount of data so far. But we focus on processes, interactions, and integration, both across trophic levels, but also across disciplines, and also moving into the Arctic Basin. And the plans we have for 2021 is to fulfill a seasonal study we have started to learn more about winter and early spring situation. And it's to extend this key transect or core transect that we are working along into the Arctic Ocean to go across the Nansen Basin and the Gäckle Ridge and into the Amundsen Basin. And this is in collaboration with SAS. And one of our main motivations for doing this is that we need to connect our regional knowledge across the Arctic Ocean. So with this plan, it's a pre-COVID-19 plan for SAS from 2019, so a lot of things may have changed. The Nansen Legacy plan to contribute with a transect, and we think that this is a very important task to do, and that SAS initiative is a very unique and important initiative, having synoptic studies that enable coupling and understanding of the different links across the Arctic, but also an understanding of the different regions. An important addition to all these cruises is that we have to improve and utilize better, especially with respect to ecosystem studies, the new technology. To combine new technology to complement remote sensing to improve under ice observation and our seasonal resolution, and that is necessary to move further in our understanding. Bioacoustic platforms like the one illustrated here from Solich and et al. Is one way we can learn more, for instance, about under ice blooms and also the glaciers under the sea ice when they can monitor who is there over long periods of time. We also need to expand our biological investigation with more sensors and use of AUV to observe more away from ships and where we work, and combined field work like done in Mosaic and what we try to aim in the Nansen Legacy and what will be conducted in SAS, prepare this ground for a more holistic understanding of how ecosystems respond to the ongoing change. So I just would like to underline this need to understand the regional systems, like we have heard about the Beaufort Gyre and that we heard about the ocean acidification and the chemistry. What are the key driver and responses for the ecosystem depends on the characteristics of these different regions. For biologists, it's also highly necessary to work across disciplines. And we need to expand our use of observational tools, but also to connect better across size groups in the ecosystem and understand more about the processes in the smaller organisms. And then to connect this to have a more pan-Arctic understanding. So we are very happy both for this webinar and the initiative of SAS and think that this is the right way forward. Thank you very much.
Thank you very much, Marit, for a very interesting presentation and also for your visionary slides at the end here. We have now open for questions for Marit, so please write them and I'll read them out. The first question is from Are Olsen. Are you aware of any long-term monitoring of phytoplankton composition in the Arctic? And if yes, are there any trends?
I think there have been the sediment trap moorings that Housegarden or Avi have had in the Fram Strait. They have collected algal material and looked at long-term trends, and they see that there are changes in composition of algae, but at the same time, it doesn't seem to be a big difference in the vertical flux of the material. But I think that these sediment traps, they can provide good information of some aspects, but not all of them. So I think that with better technology that can take samples, and this is under development, and there may be that first data starts to come, which also includes more molecular studies of water column data that is feasible. And then there is also the study by Lee from Canadian Arctic showing that there is a trend towards smaller algae in the Arctic.
Thank you. From Rubauji, if advective input is that important to Arctic production, would you expect the local production contribute little to the variability of production?
Based on the model simulation that was done in the work by Maria Varnet and her co-workers, and that was using the SynMod model, it seems like the advective component was stronger than the local production. But of course, an advected algae into the Arctic basin, and especially along the shelf break, will also need the nutrients both from the advection but also locally to grow further. So again, I think it depends on where you are in the Arctic. Maybe along the shelf break the advection will be more important, but in the central basin and maybe on the non-inflow shelf, the more interior shelves, the local production will be more important.
Interesting. From Frans Mutter. If grazing over inflow shelves has increased, become more efficient, does this mean that increase in chlorophyll A actually underestimate increases in production in these areas?
I'm not sure if we can say that grazing has been more efficient and has increased. I think that the grazier community, at least for larger copepods on the inflow shelf that I know the best, there has been a change in the community composition towards the more smaller subarctic species with more advection and warmer water. And if the the grazing is increasing, that may be that it's shifting from a larger community towards a smaller one, which are more efficient. But I missed the last part of that question. If it means that we underestimate.
See, does this mean that underestimate increase in production in these areas?
I think that estimating the production is challenging because a lot of the production estimates is from satellites, and you use algorithms to convert the biomass of chlorophyll to production. And depending on the size and the growth rate of the algae, that may change and not be captured by the algorithm. So I think that we need to combine the satellite measurements with studies in the plankton community and production measurements to actually see if we are doing okay in that sense.
And maybe you can keep your answer a little bit short here, but it's a general question from Jean-Pierre Gottisot. You mentioned shelf studies as a part of the Nansen legacy. Could you elaborate a bit?
The Nansen legacy is addressing the Northern Barents Shelf, which is an area which has undergone quite a lot of change over the past decades. So we have put together a team of 10 institutions, about 200 scientists, to investigate the geology, the benthos, the chemistry and biology and physics of the water column, and the physical processes and the atmosphere, combining fieldwork, remote sensing, and modeling. There is a web page for the Nansen legacy which could be looked into. It's nansenlegacy.org.
Okay, and then there's Alison McDonald. Can satellites see under ice blooms? If not, does this affect the conclusions concerning the importance of advection?
I think at present we cannot see through sea ice with satellites, but there are work to both try to model them, to be able to see how they develop, if they are initiated by advection, which has been suggested from some studies by Arrigo and co-workers, or if they are locally initiated, as was suggested by Philip Osmi and co-workers. So I think at present we cannot see them with satellites. We have observed them, and we don't know how frequent they are, and we don't know how largely distributed they are. So I think we have a lot to do here.
And then there's one more question from Bengt Karlsson. How would the reduction in multi-year ice influence the size structure of the phytoplankton community? Nutrient input from deep water? ?
The reduction in multi-year ice impacts the stratification in terms of freshwater additions. So that is a physical question. And then I think it depends on where you are in the Arctic. On the Pacific side, the stratification is stronger, and it's less nutrients that are brought up through the to the upper layers, and not even in the winter there is a fully convected water column. And on the Atlantic side, the Atlantification has decreased stratification and also deepened the mixing, providing more nutrients. So in terms of decreased multi-year ice, and you have different effects in different regions, and with more nutrients we could also expect to have more larger cells during bloom situations at least. But we don't know yet whether autumn blooms are initiated by mixing new nutrients, as far as I know.
Excellent. Thank you very much, Marit, for answering all those questions, and also for a very nice talk. I think we've come to the end of this webinar, a little ahead of schedule, which I think is good. no technical at this webinar, and have a beautiful day. Thank you.
Thank you.
Thank you.
Thank you.
Himlen.